Hi and welcome to this video on transpiration in plants. Transpiration is a loss of water vapour from the stomata in the leaves of plants by evaporation and diffusion. So water goes into a plant through these root hair cells, which are the outermost cells of a plant in the soil. They have this hair-like projection that maximises surface area for water absorption. And water enters these cells by osmosis, down its concentration gradient, and from there it will move into further root cells through the root organs themselves until they finally find themselves in the xylem tubes. So these xylem tubes or xylem vessels are embedded within the stem of a plant, and water will travel up through these sort of dead hollow tubes all the way up through the veins that you would find in a leaf. So the veins are made part of xylem, part of a different type of vessel called phloem, which has a completely different role altogether. And once it gets to the leaves, it can then be used as a reactant in the photosynthesis reaction. However, there is one other key reactant for this reaction, and that is carbon dioxide. For that to enter the plant, you need to have holes in the leaves themselves, and these holes are called stomata plurally. A single hole itself would be called a stoma. And as you can see in this diagram here, each stomata or stoma is flanked either side by two kidney bean shaped cells, which are called guard cells, and these can vary in their size to effectively open or close the stomata depending on the environmental conditions. So carbon dioxide can get in, but we have a conundrum because water can also get out. It could easily leave the leaf through these stomata. And if you think about what a leaf is designed to be, it's designed to be a solar panel. Nice and thin, nice and broad and flat, so that it can access as much sunlight as possible. That sunlight will carry lots of heat energy, and all the water molecules inside that leaf will be gaining so much energy from the sun that it will overcome the forces of attraction between the water molecules, causing it to turn from its liquid state into water vapour. And once it's in water vapour, it can leave through the stomata. So plants are at this kind of crossroads between making sure that they're getting carbon dioxide in but not losing the precious water that they need. Essentially, if a plant is facing kind of drought and water loss, you'll notice that plant will start to wilt and the leaves will start to flop over. This is actually a survival mechanism because when the leaves flop over, they're no longer as broad and flat as possible, so they're not getting as much sunlight, and also stomata are kind of hidden away and their access to the air is significantly reduced. However, despite the fact that the stomata can accelerate the loss of water from a plant, you also need the water molecules to evaporate from a plant to drive the movement of water from the ground. And this is called transpiration. So when one water molecule turns into water vapour at the top of that leaf and then leaves through the stomata, it creates a void, a deficit that needs to be filled by the water molecules behind it. So like a queue of people at a checkout, when one person has paid for their goods and have left that queue, the queue moves forward. So all those water molecules will move forward and upwards. And it's this particular mechanism that will allow a tree that's 50 metres or more in height to be able to defy gravity and get that water from the ground all the way to its upper surface. So we could investigate the effect of various factors on transpiration and the loss of water from a plant using this piece of equipment, which is called a potometer. And all it is a piece of plant material placed into a sealed unit containing water that has to flow through a very thin tube called a capillary tube past a scale or ruler. And you will see that there's this little air bubble here. To get this into this sealed unit, you simply need to create a gap between the end of this tube and this water level here, just for a split second, and the air bubble will be introduced. This is actually quite fiddly, and you need to line that air bubble up as best as possible with 0 to start with. If that's not possible and it's slightly off, maybe by a millimetre or so, that would be called a 0 error, and you would need to factor that into your measurements and your recordings. So if it's 2 millimetres off the 0 point, whatever reading you jot down, you would have to subtract 2 millimetres from that reading to compensate. You have here a reservoir of water so that you can repeat the practical again by replenishing the water to move the bubble back to the starting point rather than have to reset up everything from the beginning. So how is this used? Well, essentially, this plant here will be carrying out photosynthesis and will have stomata open to allow carbon dioxide in, and during that point, water will be able to escape and evaporate, often through the underside of these leaves. In which case, water will be drawn up through this system, and you can track the movement of that air bubble to show how quickly water is being taken up by this plant. Over the course of the experiment, this water level here will go down. So in an investigation, you want to have some variables, and there are certain things that will speed up transpiration by either affecting the evaporation of water or the stomata itself. So first of all, we have got light intensity. Now that can actually affect both. So with high light intensity, you get more photosynthesis. So an increase in photosynthesis. I'm just shortening that here. And if photosynthesis is happening, more carbon dioxide is needed. So the stomata are going to be open. And the guard cells that actually control the closure and opening of the stomata are sensitive to light levels. So they will open during daylight hours and close during the night, when photosynthesis simply cannot happen because there won't be any sunlight energy. And as stomata are open, it's more likely that water will evaporate out through those stomata. Another factor that could affect the rate of transpiration is temperature. Now this mainly just affects evaporation and will increase evaporation because more heat energy is absorbed by the water molecules, giving them enough energy to overcome the forces of attraction that normally keep water molecules together so that they can turn into a gas state and form water vapour that can evaporate through the leaves. Humidity, that actually will reduce transpiration because the air around the leaves will be saturated with lots of water molecules, and essentially you need to have a diffusion gradient for water to actually leave through the stomata. And if the air is already sort of full up with water itself, then there won't be any room for the water that is in the leaf to move into. So the water will stay in the leaf and won't escape. So in very humid air, there will be a low level of transpiration, and that air bubble simply won't really move. Another factor is wind movements. If it's very windy, all these water molecules will be blown away from the leaf itself, leaving a very low concentration of water in the air around the leaf, meaning that more water will leave by diffusion from the leaf itself. So wind movement will increase evaporation as well. And the last factor, I'll just squeeze it, I guess, at the top here, is surface area. As surface area increases, there will be more stomata that are available to allow water to escape, and therefore there'll be an increase again in the evaporation of water. Therefore there will be an increase in transpiration and that bubble will move faster. To vary the surface area, you could quite simply just take off leaves one by one to reduce the surface area, and you should find a decrease in the speed that this bubble moves as you remove each leaf, because you're removing all the stomata associated with each leaf. So when it's very, very light, it's very warm, and it's very windy, and a plant has loads and loads of leaves, a huge surface area, transpiration will be much faster. Daylight triggers these guard cells to open the stomata. The objective is to get as much carbon dioxide in whilst there's light available for photosynthesis. If there is a sudden loss in water availability, the cells actually lose water, and when they lose water, they flatten and straighten, and as a result they close the stomata, they close the opening of the underside of the leaf, and this is an adaptation to reduce water loss. However, in times of plenty, the guard cells fill up with water, they become turgid, and due to the fact that the cell wall on the inside of the guard cell is thicker than the cell wall on the outside, it causes the actual cell to curve when it fills with water. and that will allow the stomata to open. Another adaptation of plants in terms of guard cells and stomata is that you find the majority of them on the underside of the leaf. This is because the underside of a leaf is often very shady and will be a lot cooler than the upper surface of a leaf, and it is more likely that water will evaporate from stomata on top of the leaf than below it. Similarly, depending on the environment that the plant is adapted to, there will be this thick, waxy, waterproof layer, and you will find that plants that live very hot, dry conditions would have very, very thick, waxy cuticles to prevent water being lost. So that just about sums up this video on transpiration. Make sure you have learnt the definition for transpiration, that you are able to state the factors that could influence it and speed up transpiration, but also be prepared to explain why they have that influence on transpiration. Thanks for watching.